I'm going to explain about immunoelectrophoresis. It's another precipitin reaction technique to demonstrate the purity of the antigen or the antibody, which can be checked by this procedure. Now, the antigens or antibodies are generally purified for the purpose of developing a diagnostic kit, or the antigen may be purified for the purpose of vaccine production. So, in that situation, one should know whether the antigen sample that has been prepared or extracted is pure enough against a corresponding antibody. Similarly, the antibodies can be purified, and those antibodies after purification technique can be tested for purity, and it would be essential to have a pure antibody while using it in a diagnostic kit for the testing of the samples. Okay. The immunoelectrophoresis involves two different procedures. First, the electrophoresis of the sample. Second, the diffusion, immunodiffusion, where you make the antigen or the antibody to diffuse with one another and form the precipitin band. Okay. The electrophoresis involves the regular supply or a normal supply of a controlled power using power supply. And these are the tools that you require for the immunoelectrophoresis, which was explained earlier. A clean glass slide to be prepared with the agarose, and you need agarose of high quality grade which dissolves easily in buffer, and you need the test antigen and antibody samples. Agarose is dissolved in appropriate quantity of buffer, and you need about 2% agarose dissolved in the buffer and heated gently in a moderate temperature. After it melts completely without any particles, it is ready to be used for the preparation of slide. One can prepare a large glass slide as it was shown for single radial immunodiffusion or for the rocket immunoelectrophoresis. And that large slide can be used for running many samples simultaneously in different regions of the slide. Or one can use a simple microscope slide on which you have to pour about 3.5 ml of agarose. And that agarose is allowed to solidify after pouring it on the slide as demonstrated earlier. And once the slide is prepared, it is supposed to be kept in the moist chamber for the agarose to solidify properly and the distribution of the ions, even distribution of ions in the slide. And it was already explained how to prepare the moist chamber by using the bedding of the filter paper, which is moistened with the distilled water. And in that moist chamber, you keep the slide for about an hour, sorry, half an hour. And after that, the slide is ready to be used for the electrophoresis purpose, for which you have to punch the slide. The slide is kept on the stencil and two wells are punched for the loading of sample. One well can be dedicated for the normal pure antigen to be loaded, or in another well you can load the patient sample or the test antigen sample. So once the slide is punched, the antigen is loaded in the slide into each well, and the antigen is generally mixed with the tracking dye, that is bromophenol blue, because when you carry out the electrophoresis, it is necessary to know the mobility of the sample, and you should allow the sample to move towards the other side, and the antigen side is connected to the negative pole. Once the sample is loaded, it is kept in the electrophoresis chamber on the platform of the electrophoresis chamber such that the antigen loaded side is connected to the negative pole, and a gauze piece dipped in buffer is slowly laid on the side of the side so that it supplies the current. Both the sides are connected with the gauze piece dipped in the buffer, and the electrophoresis is run until the tracking dye comes to the other end, as seen in this figure. Once the tracking dye has come to that point, take the slide, put it on a stencil using a sharp blade, cut the trough in the middle as indicated in the stencil. After cutting the agarose with a sharp blade, lift the agarose and create the trough. After creation of the trough, the slide is ready, where in the trough you are supposed to load now the antibody. Suppose if you had taken the normal human serum in one well and in another well the patient's serum. In the middle trough you will be loading anti-human serum, and the anti-human serum is called comb serum. This is to test in the patient whether he is lacking any serum proteins against by comparing with normal human serum. The slide, once the antibody is loaded in the middle in the trough, is supposed to be kept in the moist chamber, and the result will be visible next day, that is after 24 hours after the immunodiffusion takes place. So here is an example of the purity of a particular antigen. The antigen was loaded in both these wells. One, the known pure antigen, let us say in the upper well, and in the lower well, the purified antigen, which is experimental. And then after the completion of electrophoresis, the electrophoresis was run in this direction, that is left to right. The antigen loaded well side was connected to negative pole. So as the tracking dye moved to the end of this slide, the trough was cut in the middle after the completion of electrophoresis, and the antibody corresponding to that antigen was loaded in the central trough. In this slide, it is very clear that the pure antigen, when it is compared with the experimental antigen, which was isolated or purified, both of them are pure enough. The pure antigen compared with the isolated one shows that it is pure enough like the pure antigen. So from this technique, you can prove that the technique that is adopted for the purification of the antigen is quite good and you have achieved the purity that you desired. Okay. In this slide, you can see the series of different antigens were loaded in different wells and they were run at the same time by electrophoresis. But in the central well, different antibodies were loaded. The first part, that is the uppermost part, shows NHS stands for normal human serum. That is in this well, on the upper well, normal human serum was loaded, and in the lower well, patient serum was loaded. Now, this is to compare whether the patient has all the protein components that are supposed to be present in the serum of a normal person, or the patient is lacking any of those proteins. For that reason, in the central trough, anti-human serum is loaded, that is Coombs serum. Here you can see a large boat-shaped band which is appeared in the positive side or the positive pole of the slide, which has moved faster, and it is supposed to be of the albumin, human serum albumin. Here also on the lower side you can see large boat-shaped band. Boat-shaped band is the characteristic of albumin, and albumin has the fastest electrophoretic mobility. Rest of the other serum components, human serum components, have very poor electrophoretic mobility at that given pH, and pH of about 8 or so. Here you can see in the slide the IgG band, human immunoglobulin G. IgG band is very close to the well and it is still on the negative side. It has not moved towards the positive side. It has a very poor electrophoretic mobility. This is of the IgG. Immediately next to that, this band is of the IgM, human immunoglobulin M. Then comes very close to the well will be IgA. And that can be proved here in the second diagram of the slide, where anti-IgG is loaded. Anti-IgG is the immunoglobulin, human anti-immunoglobulin, which is produced in another animal by injecting a pure human immunoglobulin G. Let us say that you had purified immunoglobulin G by using ammonium sulfate precipitation method and then column chromatography method. So once you have purified that immunoglobulin and column chromatography using gel filtration. Now we want to test whether that IgG that you have isolated from the human serum is pure enough, which can be compared with already purified human IgG. Here is the example where the test sample is loaded in the lower well, and in the upper well you have loaded the pure human IgG. Okay, and you will see that when you add the anti-IgG in the trough, the anti-IgG will react with human IgG, and if it is pure enough, you will get only one band of IgG. This shows that the purification technique that you had adopted to purify IgG from the serum sample of the human is good enough. Similarly, in the second another slide, you can see the purity of IgM by adding anti-IgM. Similarly, IgA. Then here in the lowermost part, they have shown kappa and lambda chains. These are the chains of the immunoglobulin light chains. Kappa chain and the lambda chains are the immunoglobulin light chains. One can purify those light chains also after separation of these light chains from the heavy chain, and purity of these light chains can be tested against the antibody developed against these light chains. So this is how immunoelectrophoresis is used for the checking of purity, whether it is an antibody or an antigen. When you are checking the purity of an antibody, it is necessary to have antibody against antibody. And if it is human IgG or human IgM, human antibody, one can raise the antibodies in other animals. And that antibody can be used for testing the purity of the human antibody which has been purified. And pure human IgG or purified antibodies are generally used for diagnostic technique. They can be labeled with radioisotope, or they can be labeled with the enzyme, all right, or they can be labeled with fluorescent probe and then used in the diagnostic kit for the detection of a particular antigen or for the detection of the antibody. Okay, and this immunoelectrophoresis technique is a very useful technique to test the purity of the samples where the appropriate techniques have been used for the purification. Thank you very much.